Hereditary spherocytosis is the most frequently encountered inherited intracorpuscular hemolytic anemia in white populations and follows an autosomal dominant pattern. Red cells lose their normal biconcave disc shape and become spherocytes, small, dense, sphere-shaped cells that are selectively trapped and sequestered in the spleen, making splenomegaly a prominent finding. These spherocytes show increased osmotic fragility, meaning they lyse more easily than normal cells when placed in a hypotonic solution. A characteristically elevated mean corpuscular hemoglobin concentration, MCHC, is also seen. This is unusual because most anemias either lower or leave unchanged the MCHC, making its elevation here diagnostically helpful. The molecular basis involves defects in spectrin, ankyrin, protein 4.1, or other red cell membrane skeletal proteins, with spectrin deficiency being the most common final pathway. Hereditary elliptocytosis, another autosomal dominant condition, produces elongated, oval red cells. It can be associated with hemolytic anemia and splenomegaly, but many affected individuals remain clinically asymptomatic. For enzyme deficiency anemias, G6PD deficiency is the most common form. It is X-linked, meaning it predominantly affects males, and occurs in approximately 10% of African Americans as well as people of Mediterranean origin. G6PD normally protects red cells from oxidative damage. Without it, exposure to oxidative stress from infections, drugs like primaquine and sulfonamides or fava beans, triggers acute intravascular hemolysis with hemoglobinemia and hemoglobinuria. The blood smear may show bite cells, which are erythrocytes that appear as if a chunk has been taken out of them. The anemia is episodic and self-limited. Pyruvate kinase deficiency is the second most common enzyme deficiency hemolytic anemia. Unlike G6PD deficiency, it follows autosomal recessive inheritance and causes hereditary non-spherocytic hemolytic anemia that is chronic and sustained rather than episodic.